All right, so this is the Osmosis Lab Quiz practice video, and we're going to go over basically the kind of structure and material that's going to be on your Osmosis Lab Quiz. So before we go into that, some of you have already shown me that you understand the hyper, hypo, and isotonicity comparisons, and we're just going to review it a little bit before we get into this lab quiz practice. So as you can see on this left side over here, for tonicity, there are three different descriptions of a solution surrounding a cell. And so the first one would be a hyper or hypertonic solution. So that means inside of the solution surrounding the cell, there's more solute, whether that's salt or sugar. It's a saltier or more sugary solution on the outside of the cell compared to the inside of the cell. So if there's more solute, it means that outside of the cell, there is a higher concentration of solute, which means that the water is going to move wherever it's salty or wherever it's sugary. So because the inside of the cell is less salty or less sugary, the water will move from the inside of the cell to the outside of the cell. And then the cell is going to lose water, it's going to wither, it's going to shrink, it might even crenate and die. There's a second type of solution, which is a hypo or hypotonic solution. And just keep in mind that the pronunciation or listening carefully for the difference between the two is super, super important because they sound kind of similar because they're basically the same word except for like the difference of one or two letters. So a hypotonic solution outside of a cell has less solute. So it's less salty, less sugary, or just more watery in general. So the outside solution has a lower concentration of solute. And so water is going to move from the outside solution into the cell because the outside solution has more water, so it has more water potential than the inside of the cell. So water is going to go from high to low, and water will do osmosis and go from the outside of the cell into the cell. And if a cell gets too much water, like an animal cell, it'll expand until its capacity, and then it will burst or lyse. But plant cells, remember, plant cells don't burst because they have a cell wall. The third type of solution is an iso or isotonic solution, which means that the concentration of salt or sugar or starch or anything on the inside of the cell and the outside of the cell are exactly the same. That means that the water is going to be moving inside and outside of the cell at the same rate. So if two water molecules move into the cell, then two water molecules that were originally inside of the cell also got to move outside of the cell to maintain the similar concentration. And hypertonic and hypotonic solutions are just situations of cells in solutions before they reach an isotonic kind of situation. So being isotonic or being in balance and having the same concentration of solute inside and outside of a cell is always the goal. Hypertonic and hypotonic solutions are just solutions that haven't reached isotonicity yet. Or if cells are doing active transport and making sure that the cell never reaches isotonicity, because in some situations in bodies that is required. So we're going to go over first the types of cells here, and then we're going to go over the net movements of water based on the arrows. And again, as you can see, if water is moving in and out of an isotonic solution inside of and outside of a cell at this equal rate, then the net movement is going to be considered zero, right? If one moves in, one moves out, they cancel out. Basically, it's like having zero move, but you want to make sure that you understand that it's because things are moving at equal rate, not because nothing is moving all of a sudden, because molecules are always moving if we are above 0 degrees Kelvin or negative 273 degrees Celsius. Okay, so up here first, we can see that we have over here cells that do not have a cell wall, which would be animal cells. And then this outside layer right here of a cell is known as a cell membrane, right? And then this dark center part that contains the genetic material of a cell is known as a nucleus. Over here, we have a cell that is bursting from being overfilled, and that would be called lysis, right? Remember, to lyse means to break. And over here, we have a cell that is losing a lot of water, and therefore it is shriveling or shrinking, so it is crenating. Okay, over here, we have a different kind of cell, and these cells have cell walls, which means we have plant cells. And the thing in the center of the plant cell, remember, that helps them keep water content or keep their turgor pressure is called a we'll call it a central vacuole right here. Inside of plant cells, we still have a nucleus because they are still eukaryotic cells. We also have these organelles that make them green and help them do photosynthesis, which are chloroplasts. When you have a plant cell that is super filled with water, but it's not bursting because there's a cell wall and the central vacuole is helping keep the pressure maintained. We call this cell, the super filled, sturdy cell, a turgid cell. And here we have a cell wall, which is the outside layer of a plant cell. And here, right inside of the cell wall, you can see that even though the very outside layer of a plant cell is a cell wall, right inside of it is the cell membrane still. And also this is kind of important that you can see that there's a keywords list over here that you'll need, and the words can be used more than once, except for kind of the section that we're going over right now, which is like the descriptors. And over here, when the cells shrink, kind of like the way that animal cells do, but because there's a cell wall that provides some support, the cell membrane will still be attached at certain points to the cell wall. And so this is a plant cell experiencing plasmolysis. So the plasma is kind of shrinking. The cytoplasm is shrinking in because there's not a lot of water in the cell, but it's not completely withering and dying, right? Because if you pour water onto a plant that is a little bit withered, it can rejuvenate and fill its cells again with water and end back at being turgid or just kind of, uniform. Over here, we're going to go over now different tonicities based on the arrows of water movement, and we're going to be telling kind of the general direction of the movement of water. So we're going to first look right here and kind of check out, what's happening with the arrows here. So we have an arrow that's going out, so one water is leaving, and then we have an arrow that's going in. So we're actually just going to, let me change the color on this, make it stand out a little bit more. So one in and one out can kind of cancel with each other because it's basically zero net movement if one goes in and one goes out. Here's the same thing. So one's going in, one's going out. So what is our net movement? Are there any arrows left? The net movement of water is, in this case, it is, hold on, let me change this. The net movement of water is going to be 0 here because we canceled out all the arrows. And so if there's a net movement of 0, there's going to be an isotonic solution. Yeah, bear with me as I write with my mouse. Okay, over here we're looking at another solution that this cell has been placed into, and as you can see, the arrows are slightly different from over here. So we want to cancel them out again. So an arrow going out is going to be canceled out with an arrow going in. But we look at these other two arrows, and you actually can't cancel them out because they're both going in. So now the net movement of water is no longer 0. Right? The net movement is into the cell. And hence, there's a lot of water going in, and an animal cell cannot take too much water, otherwise it will burst or it will lyse, right? So if a lot of water is going into the cell, it means that the outside has a lot of water, and the inside of the cell is salty, right? So the outside has a lot of water, and it's not as salty, then it's going to be a hypotonic solution, because it is a less solute or lower concentration of solute on the outside, right? There's more water on the outside than salt or sugar. Over here, let's do the same canceling out. So a water going in and a water going out can be canceled out. However, the last two arrows cannot be canceled out. And so the net movement of water follows these arrows, and it's going to be out of the cell, right? The water is moving out of the cell. It's because the outside of the cell is saltier or more sugary than the inside. Okay? The outside is saltier. The water from the inside is going to try to balance that out and leave to make the water outside, to make the solution outside more diluted, so that eventually they will be isotonic. So as the water leaves to go to the outside, because the outside has a lot of solute or too much or higher solute concentration, we're going to call the outside solution hypertonic. Okay. We're gonna go down here and look at the same thing now to see what we can get out of here. So if we have a water going in, we can cancel that out with a water going out, right? Because that's technically 0 net movement. Over here, we have an out and an in as well, so we'll cancel these out. And then you'll notice that afterwards, there are no other arrows, so therefore our net movement of water is 0. And you know that a net movement of water of 0 means that this is an isotonic solution. So basically, the solution matches the contents of the cell in saltiness. Over here, we're going to look at this turgid cell, and so we're going to cancel out a water moving out with a water moving in. But then our arrows left are saying that the water is going to be net movement into the cell. And a net movement into the cell means that the outside is less salty than the inside, and it's trying to balance the inside by adding more water to it to make it less salty inside. Okay, so the outside is less salty, it has a lower concentration of salt, therefore it is going to be Hypo. Remember, lower concentration is Hypo, low and O. Okay, and then in this plasmolyzing cell, we're going to talk about the arrows. So you're canceling water leaving with the water going in, but then the arrows left over show that the net movement of water is going to be out of the cell. And when the net movement is out of the cell, it's because the outside of the solution is salty and it's trying to balance out with the water inside of the cell to dilute the outside solution to make it eventually also less salty. So if the outside is too salty, it's a high concentration of salt, and therefore the solution outside is hypertonic for salt when compared to the inside of the cell, at least. Okay, again, these are all relative. You're comparing the outside solution to the inside of the cell. It's relative to that specific cell, right? A different cell could be hypo or hypertonic in the same solution, just depending on the actual concentration contained inside of the cell. So let's talk about the other side of this practice now. So we're looking at the dialysis tubes in cups like we did originally in our diffusion part 2, right? So here we have a solution that has pure water inside of the dialysis tubing, and we have a 5% salt solution. So pure water, you're gonna wanna make sure that you understand that pure water means that we have a 100% water, okay? And over here, if we have 5% salt, that means we still have 95% of it being water. Okay, so we're going to be comparing now. And over here, you want to know that it's 0% salt. Wow, sorry, my handwriting is so bad. So when it's pure water, it's 100% H2O or water and 0% salt. When they give you a 5% salt solution, you know that's 5% salt, and then the rest is going to be water. So we're going to be comparing the level of salts now, right, for hyper, hypo, or iso. So the outside solution is going to be, this outside solution right here, is going to be hypertonic. And you ask me, why is it hyper? Well, it's because outside, oh, good spelling, my bad. Because outside of the cell, or outside of the dialysis bag, we have a higher concentration of salt, right? 5 is higher than 0. Therefore, the outside solution is hypertonic. If it's hypertonic, that means the net water flow is going to be out of the dialysis tube, right? The water is going to go where it's saltier. And inside, it's not salty at all. Remember, there's 0 salt. But outside, it's saltier because there's 5% salt on the outside. So the water is going to leave the tube to go outside. And then after water has left the tube over a period of time, the tube will weigh less because water is leaving, and water is what is causing the mass inside of the tube anyways, right? Because originally it's just a tube of pure water. Over here, 25% salt and 25% salt. You probably already have a good idea of what's going on, but we want to make sure that we clarify that what the rest of the solution is made out of, right? 25% salt, which means that we're going to have 75% H2O. Over here, same deal, right? It's pretty obvious what the answer is going to be just because the numbers already match. So you should already know in your head where this is going. So when you compare these two solutions, 25% salt compared to 25% salt, they are equal, which means they are iso, right? Outside solution is going to be isotonic to the inside because, again, both of them are 25% salt solutions, which means that the net flow of water is into and out of. So you can write into and out of. But ultimately, in and out means zero net movement, right? Because the in and out cancel. So it's more correct for you to write zero net movement because it's asking for net flow right here, okay? But writing in and out is not incorrect, remember, because molecules are always moving. So if the water that's moving in is also matched, or sorry, out is also matched by water that's moving in at the same exact rate, then the tube will weigh the same. I'm gonna have to fit the same right here, right? Because the water moving in is being matched by the water moving out. So after you could wait like days, you could wait years, technically this would have the exact same mass at the end of that time period. So here we're going to do our last comparison now. 20% salt means that there is about 80% that is the solute, which is going to be water, right? And then for 10% salt, the rest of the solution is going to be the 90% of the solute, which is water. And we're only doing this for clarification purposes if you can't really understand which one has more water and why water is moving from high to low concentration, right? So 20% inside and 10% outside. That means that inside only has 80% water and then the outside has 90% water. Okay, so the concentration of the solution on the outside, right, the outside solution, is lower of salt than on the inside. If it's lower, that automatically means it is a hypotonic solution. Okay, and then we're going to talk about the net water flow. So remember, water always flows from a higher concentration to a lower concentration. So between 80 and 90, which one is higher? Right, 90 is higher, which means that water from the outside is going to flow into the cell because the concentration of water outside is higher. There's a higher water potential than there is on the inside. That's it, right? So our net flow of water is going to be into the dialysis tube. And if you keep adding water to the tube and it keeps gaining water and growing in amount of water, then eventually the tube will weigh more after equilibrium is reached because you just keep adding water to it because remember, they're trying to equalize to reach equilibrium and be the same concentration. So eventually, if you really let it and the dialysis tube had enough room in it, they would both reach about 15% salt solution and be the same concentration, and they would be isotonic. So that covers our quiz review. This is in preparation for on Thursday when you are completing your osmosis quiz on your own, and then you are going to go ahead and complete FRQs on membrane transport.